Hello, and welcome to our informational video on our company's innovative SNP chip genetic test for breast cancer, focusing on the RS1800057 variant, also known as P1054R. We want to provide a better understanding of the genetic basis for breast cancer and explain our new revolutionary test. Today, breast cancer affects approximately 12% of all women in the general population, with an estimated 1 in 8 women developing it at some point in their lives. While breast cancer can be influenced by lifestyle choices and environmental factors, genetics also plays a key role. In addition to the BRCA1 and BRCA2 genes, the TP53 and ATM genes are linked to increased breast cancer risks as well. These genes are crucial for maintaining DNA integrity by repairing damage and controlling cell growth. With mutations in these genes, there is an increasing risk of cancer due to impaired tumor suppression and DNA repair mechanisms.
Today, one of the most well-known tests for breast cancer is 23andMe's BRCA1 BRCA2 report. Among the variants tested is C5266-C or RS803-57906, which is a very impactful mutation that has a max score of 6 on Snipedia. The duplication is found in the BRCA1 gene, which is expressed primarily in breast and ovarian cells. This duplication causes a frameshift mutation that leads to a dysfunctional DNA repair protein that can't regulate the quickly replicating cells. It makes sense that this leads to breast cancer, as errors will accumulate more quickly, leading to uncontrolled cell growth. However, their test is severely limited, as only 44 of the over 4,000 genetic variants in those two genes are tested. Additionally, it does not include any variants from the other dozens of genes that are known to contribute to breast cancer risk, such as TP53, CHEK2, and ATM. Our SNP chip test offers a much more comprehensive screening, covering not just the SNPs that are tested by 23andMe, but also the variations from the other 3 genes. One of our key variations is RS1800057 from the ATM gene. The RS1800057 variant is particularly relevant because it has shown a substantial impact on breast cancer risk. When this variant is present, it disrupts the ATM protein's ability to repair DNA, leading to an estimated 27% increase in breast cancer risk. The ATM gene is expressed in virtually all cells, and similarly to RS803-57906, it makes sense that a variation in this gene could lead to quickly accumulating errors that lead to an uncontrolled cell growth in the breast area.
Our SNP chip test offers a more thorough analysis of breast cancer risk compared to 23andMe by examining a broader set of important genetic markers. In addition to key variants in BRCA1 and BRCA2, our test also covers other crucial genes related to cancer risk, such as TP53, ATM, and CHEK2. Research shows that including these additional markers significantly improves the accuracy of risk assessments. For example, pathogenic variants in TP53 are associated with a 72% lifetime risk of developing breast cancer, while mutations in the ATM are linked to a 25% lifetime increase. By incorporating these genes, our test provides a more comprehensive understanding of an individual's genetic risk for breast cancer. To avoid issues like those faced by 23andMe, we are committed to clearly explaining the limitations of our SNP chip test. While it provides a comprehensive analysis of breast cancer risk, not all genetic variations are currently identified. For example, variants like the RS1800057 may indicate an increased risk but don't guarantee breast cancer development. These genetic variants may only slightly increase risk, and the actual risk varies based on individual factors. Breast cancer risk can also be influenced by lifestyle, family history, and environmental factors as outlined by the National Institute of Environmental Health Sciences. A positive test result suggests higher likelihood, not certainty. For example, variants in genes like TP53 are linked to a 72% lifetime risk, while ATM mutations have a 25% lifetime risk. However, many people with these variants may not develop cancer. Additionally, high-risk results can cause emotional distress, especially if no symptoms are present. Genetic information is so sensitive and can affect well-being and insurance eligibility. So we prioritize privacy and adhere to strict privacy regulations to protect your data. We want our customers to understand that genetic testing is just one part of the picture, and both genetic and environmental factors should be considered when assessing risk.
Thank you for watching and listening to our S&P CHIP breast cancer test design informational video. We hope that this video has taught you a few things on the benefits of S&P CHIP testing and the genetic causes for breast cancer and the threat that they may pose to you. Providing you with a more in-depth procedure for our test is our goal, and we hope that we have caused you to consider if S&P CHIP genetic testing is right for you.